Good day, everybody. It's my pleasure to be here today as President and Scientific Director of the Gairdner Foundation to introduce this year's Gairdner Lecture at CUGH. The Gairdner Foundation was founded in Toronto in 1957 by Jim Gairdner, a philanthropist, who wanted to recognize those researchers whose research has had impact on human health. We celebrate excellence with the Canada Gairdner Awards. And over the years, we've given out over 380 such awards, 95 of whom have gone on to receive Nobel Prizes. Amongst those awards is the Global Health Award that we're celebrating today. And of course, all those laureates make a fantastic network of leaders that we convene at symposia, lectures, and events like this one. And we hope to use this celebration of excellence to inspire the next generation of scientists and innovators. One of the awards that we give is the John Dirks Canada Gairdner Global Health Award. This has not been around since 1957. This was the brainchild of the previous president of the foundation, John Dirks, who really had a passion for global health research. And so in his honor, we have named this award in his name, and it's awarded every year to scientists who's outstanding achievements in global health research that has impacted on global health in the developing world. This year's award, 2021 award, has been adjudicated but not yet announced. It will be announced on April 7th, and we will be bringing the 2021 laureates to CUGH next year. We encourage you to go to our website and look into the announcement. You'll see it on YouTube at that time, but also consider nominating great scientists and researchers whose research you admire that's had impact on health worldwide through our nomination portal, which will be open soon after that in the middle of April. Deadline October 1, 2021 for nominations for the 2022 awards. So about 10 years or so of global health awardees, I thought it would be fun to just see what they're up to today. We're living in COVID pandemic times, and we know that so many people have pivoted what they've been doing to have an impact and use their skills to impact on the COVID situation. And it didn't take long for me just to do a quick browse through the literature and find some well-known names that many of you will know who have contributed now to the COVID situation, COVID pandemic, using their skills in different ways. The Abdool Karim you're going to hear from later, presenting the South African response. Cesar Vitória, working on maternal and child health, has been doing SARS antibody prevalence in Brazil, where Brazil has been extraordinarily badly hit by the COVID pandemic. Chris Murray, using their data sources to do predictive modeling of COVID. Tony Fauci, most famously perhaps being the trusted voice of science in the US in the COVID situation. And then of course, King Holmes, Peter Piot, using their experience in Ebola and HIV to develop lessons for COVID. And Quarraisha working with the WHO on repurposing antiviral drugs. Just some of the many ways that our laureates continue to make an impact worldwide. So without further ado, what I want to do is to introduce the 2020 John Dirks Canada Gairdner Global Health Awardees, who are Professor Salim and Quarraisha Abdool Karim. They were awarded for their discovery that antiretrovirals prevent sexual transmission of HIV, which laid the foundations for pre-exposure prophylaxis, PrEP, the HIV prevention strategy that's contributing to the reduction of HIV infection in Africa and around the world. They are both at CAPRISA, the Center for the AIDS Program of Research in South Africa, and at the University of KwaZulu-Natal in Durban. And they both also have appointments at Columbia University. So without further ado, I'd like to ask them to give their lecture, which is entitled HIV Prevention in Women, Trials and Tribulations, Quarraisha and Salim Abdool Karim.
Friends and colleagues, I would like to start by thanking the Gairdner Foundation for providing us this opportunity to share with you our body of research that spans 3 decades on preventing HIV infection in women, the trials and tribulations that we were recognized for through the John Dirks Gairdner 2020 Award.
It is an honor for Quarraisha and I to share this information with you today at the Consortium of Universities for Global Health. We hope it will convey to you the sense of excitement, enthusiasm, and the curiosity that has gone into almost 30 years of research that is being recognized by the Gairdner Foundation.
Greetings, friends and colleagues. On behalf of Salim and myself, I'd like to thank the organizers for the opportunity to share with you today our presentation in recognition of our John Dirks Gairdner 2020 Global Health Award. We will take you through a 3-decade journey on our research focusing on HIV prevention in women, the trials and the tribulations. This will be a 2-part presentation where I will start by describing the high burden of HIV in young women in Africa, the age disparity and the cycle of transmission that drives high rates of infection, and then addressing the gap in terms of the prevention challenges for women-initiated technologies. Salim will then continue on the Tanofeva gel efficacy and why we had the results we had, what is the ongoing risk in young women, and close with some novel prevention technologies in women. But first off, in the midst of the COVID pandemic, it is so easy to forget that there are many other pandemics ongoing in the world. And I want to start with this brief reminder of the global HIV pandemic and share with you the 2019 data. Globally, there were 38 million people living with HIV, about 690,000 deaths. And this is despite the fact that we have immense progress in terms of rolling out antiretroviral treatment around the world that has changed HIV from an inevitably fatal condition to one that is chronic and manageable. But of greater concern is the 1.7 million new infections that we continue to see. Another way of looking at this number is the 4,600 new infections taking place each day. And importantly, Africa still continues to bear about 70% of the global burden of HIV. In sub-Saharan Africa particularly, young women continue to account for 1 in 4 new infections taking place. Just to give you a better sense of this epidemic and the burden of infection in young women, I take you to this study done in high schools, in 14 high schools in one of the highest burden districts on the east coast of South Africa. And here you see HIV infection continues to be rare among men. But in contrast, in women, you start to see already by age 15 about twice the number of infections, and that steadily increases to by age 20, 1 in 5 women are almost infected with HIV infection. These high rates of infection in young women in KwaZulu-Natal can also be gleaned from this population-based survey undertaken in the same community where the high school study was done. And there are 7 out of the 11 health districts in KwaZulu-Natal have an HIV prevalence of more than 40% amongst pregnant women. Here we see an overall prevalence in this community of a survey conducted around that included about 10,000 people of 36%. And in women, you see an overall prevalence of 44% compared to 28% in men. And you see the steady increase following the red bars in women from age 15 to the point by age 25, every other woman is infected. By age 30, we are seeing about 70% of women infected. You see the slow rise of infection among men following the blue bars, and really taking off around age 25 to 26, where 1 in 5 men are infected. A rapid catch-up by age 31, 32, 40% of the men are infected, and by age 34, 50%. But I'm going to focus now on what is really driving high rates of new infection in already high prevalence communities in sub-Saharan Africa. And I take you back to 1990, when we conducted one of the earliest community-based surveys in Africa and published this paper that demonstrated this age-sex difference in HIV acquisition. Again, following the red bar, infections in women, the blue bar, infections of men, you see at age 10 to 14, there's very little difference in HIV infection between young men and young women. But by age 15 to 19, in an overall prevalence of less than 1%, we were already seeing 6% infection in women 15 to 19, still rare in young men. By age 20 to 24, infection peaking in women, and then you see this 5 to 7 year age difference for peak in men taking place around 25 to 29 years. Now, more recent data across Africa demonstrates this age-sex difference, and particularly in Eastern and Southern Africa, where despite a high HIV prevalence, we continue to see high rates of new infection, all attributable to this age-sex difference. The population-based survey that I referred to earlier gave us the opportunity for phylogenetic sequencing from about 90 clusters identified from about identified in the survey, and we were able to generate this schematic that confirms how transmission is occurring in this region. And of note is that young women under 25 are getting infected from men 25 to 40 years. Only 1 in 5 of these men know their HIV status, yet 2 out of 5 have a viral load greater than 50,000. And most of the infection in women under 25 is from infections in men, where the average age difference is 8.7 years. These men in turn are getting infected from women 25 to 40 years. So if you only had relationships, sexual relationships between men 25 to 40 and women 25 to 40, where the mean age difference is 1.1 years, we would have seen the end of this epidemic. The fact that we have these young women acquiring infection and advancing to age 25 to 40 ensures that there's this continuous cycle of infection taking place, and we have to break these chains of transmission and address all 3 points in terms of the cycle of transmission. These data on the cycle of transmission that was published in Lancet was also shared with globally and was adopted and influenced the Get on Fast Track UNAIDS strategy, and came back to South Africa, where in the South African national strategy for 2017 to 2022, you can see the improvement in the presentation of the schematic of the chains, cycle of transmission being adopted both globally and nationally. By now, you have a good sense of the magnitude of the problem in young women in Africa. But we have to ask ourselves the question, the same question that Zena Stein asked way back in 1990 in this commentary in the American Journal of Public Health, that all of the tools that we have to prevent HIV infection, abstinence, mutually faithful, behaviors, male-female condoms, medical male circumcision, which of these tools are for young women in Africa? So if you were a young woman, 20-year-old, having a relationship with a man, 25 years old, how are you able to negotiate safer sex with this older partner? As the epidemic grew in South Africa, and you can see the progression from 1990 through to 2010, the explosive spread in the years from 1990 to 2000, and then the stabilization from 2000 onwards, we, together with many others, continued to do microbicide trials. And microbicides are products that are used vaginally or rectally to prevent sexually transmitted infections. And we initially were using products available on the shelf and had been registered either as vaginal contraceptives. and trial after trial using surfactants initially, and then using more different products, we tested and continued to test, and each trial gave us negative results. In fact, we became quite the expert on failure. But in 2003, in providing antiretroviral treatment in our clinics, both in the urban and rural community, we came across a drug called Tanofeva. It's an effective therapeutic agent with a good safety profile that's rapidly absorbed, with a long half-life, and rapidly suppresses viral load. And we already had some evidence of its protective benefits in monkey studies. And we decided to change tack and start to look at the evaluation of Tanofeva as a potential prophylactic agent for preventing HIV infection in women. The idea of using it sexually linked or coitally linked came from the PMTCT studies, HivNet 012 at that stage, where one dose of nevirapine was given to the mother just before birth, and then a dose to the infant within 72 hours, and provided us a 41% reduction of transmission from infected mother to infant. Given that many of the women who would potentially participate in our trial were not seeing their partners on a regular basis, we were requested to provide a technology that would offer protection at the times that they were having sex. And using all of the data available, we came up with the BAT24 strategy, which is insertion of one gel up to 12 hours before sex, a second 12 hours after sex, and no more than 2 doses within a 24-hour period. Now, we proceeded with this trial in 2003, despite criticisms from some of our colleagues that the trial was doomed to failure. In 2010, the trial was completed, and we presented the data at the International AIDS Conference in Vienna, and simultaneously published that in Science. And overall, what we found was a 39% protection against HIV infection, and with 74% protection with high Tanofeva levels. And Salim will come back to this high threshold of drug level needed for protection. Significantly, this was regarded as one of the top 10 scientific breakthroughs in 2010, and also in Lancet. And it reflects the challenges that the field was facing during this time, where 37 trials later, we only had a handful of trials, 3 trials that consistently showed protection against use of medical male circumcision for preventing HIV infection, 1 STD trial showing 42% protection in Mwanza, Tanzania, and then 1 vaccine trial with some indication of protection. So since the trial results, there were many oral PrEP studies and topical agents evaluated, and in 2015, WHO recommended PrEP as a global standard for all for all people at risk of HIV acquisition. So it took nearly 5 years from the CAPRISA 004 proof of concept to the point where PrEP is now widely available and accessible, and already making huge impacts in terms of preventing HIV infection. In the conduct of CAPRISA 004, we also had a serendipitous finding, which is that Tanofeva gel also prevented HSV-2 infection. And about half of the participants in the CAPRISA 004 trial benefited from this particular indication. HSV-2 is a very common sexually transmitted virus, and this is quite a significant finding. With that, let me hand over to Salim to continue this presentation.
So Quarraisha has covered the challenges we face in HIV prevention in young women, and the journey in order to find a new technology that can empower women to prevent infection. So let me capture what has been the other side of that equation, trying to understand why the Tanofeva gel efficacy was lower than we expected, and why is it attenuated? Well, when we look at the efficacy of Tanofeva gel across the various trials, we see a wide range of outcomes. However, if we reanalyze these data based on a case control method, on the looking at women who were adherent to the gel, there we find a much more consistent benefit in the region of 52 to 57%. So why the difference? The difference really revolves around the challenges of ensuring that we can encourage women to have high adherence. So it's a behavioral issue, but it's not only behavior, it's also about looking at the biology. So one of the things that we wanted to get a sense of was, are we in a position to get some idea of what level of Tanofeva do we need to achieve in the genital tract in order to get protection? Do we have a correlate of protection for Tanofeva gel? And we did an analysis that provided us with some indication. And so it indicated to us that women who were adherent had much higher levels of Tanofeva gel, and that translated into higher levels of Tanofeva in the genital tract. And so when you see the levels of Tanofeva that were above 1,000 nanograms per ml, you can see the way in which the infection rates were very low. However, when the Tanofeva levels were below 1,000, you can see that we see very little difference with placebo. So we know that we've got to achieve pretty high levels of Tanofeva in the genital tract, and that means women have to be adherent, and if they're not adherent, then we have a challenge. And it is challenging to be adherent to the gel. So we decided to look at what besides behavior, besides adherence, is impacting. And we undertook this study, and this came as a big surprise to us. What we did is we looked at the vaginal microbiome. And in looking at the microbiome, we were trying to understand whether the microbiome was impacting on Tanofeva efficacy. And what we found was yes, that in women who had lactobacilli dominance, we found that they had a high level of Tanofeva gel efficacy. However, in women that had a much more diverse microbiome in the vagina, we found a much lower level of efficacy. And when we looked at one particular organism that is dominant when there's diversity, it's Gairdner vaginalis. And what we found was that when we co-cultured Gairdner vaginalis together with Tanofeva, we found that the Tanofeva levels dropped rapidly. There is something about Gairdner that's leading to the drug being absorbed and metabolized, but there's very little left to reach the cells in order to protect these women. So now we have a better understanding. It's not just about behavior, it's about the vaginal microbiome and the presence of Gairdner vaginalis that's consuming the available Tanofeva quite rapidly. That gave us an insight into trying to understand why these women are at risk. We now understand the microbiome is undermining Tanofeva gel efficacy, but what's increasing risk? And so we decided to look at that. And when we did an analysis of genital inflammation, and we will broadly call it genital inflammation, but it's essentially raised pro-inflammatory cytokine markers in the genital tract. In this analysis, each column is one woman. And when there's a blue square at the top of the column, that's a woman who later became infected with HIV. In the yellow columns, those are women who remained HIV negative throughout follow-up. And each row is a cytokine. And in this heat map, if you just eyeball it, you can see that on the left-hand side, where it's largely raised, that the cytokines are raised, you can see that most of the women are in the blue squares. In other words, they went on to acquiring HIV. Whereas on the right-hand side, which is largely green, the women are in the yellow squares. In other words, they are not acquiring HIV. So at a very simplistic level, we came to the conclusion that even in the presence of genital inflammation, women are at much higher risk of acquiring HIV. Now, trying to understand that risk is somewhat challenging, especially in relation to adherence. So when we looked at women who were adherent and women who were not adherent, what we're trying to understand there is, does Tanofeva make a big difference, and does the presence of genital inflammation make a particular difference? And so when we look at women with gel adherence and non-gel adherence, we find that Tanofeva makes a particular difference in women who do not have genital inflammation. In the presence of genital inflammation, you can see that the two lines are tracked with each other. In other words, little benefit from Tanofeva. So that gave us some clues that genital inflammation was playing an important role in risk, and it was playing an important role in undermining the benefits of Tanofeva. And when we look at that in a slightly different way, trying to understand what might be associated with this inflammation, what's causing this inflammation, well, one of the organisms we looked at was human papillomavirus. And in this analysis, when we look at HPV, in women who have either persisting or acquiring new HPV or clearing their HPV, remember there are multiple types of HPV, so they come and go all the time. When you have a situation where HPV infection or clearance is, or persistence is occurring, the risk of infection remains high. However, in women who do not acquire HPV or do not clear it, what we find is that they have a much lower risk of HIV. So it looks like HPV may be a contributor to the genital inflammation that is then subsequently impacting HIV. Now, why would we see this risk? What is it about genital inflammation that's increasing risk of HIV? In this analysis, we looked particularly at CD4 positive cells. And in the CD4 positive cells, what we found is that women with genital inflammation had raised numbers of CD4 positive cells. And often they were activated CD4 cells and they were closer to the lumen. So now we're beginning to understand a mechanism. Genital inflammation may be associated with HPV, but when it is present, it increases CD4 cells, and those CD4 cells are primed to receive for infection when HIV comes along. And so now we're having a better understanding about the underlying mechanism. So how does all that help us in developing novel HIV prevention technologies? Well, for us, after almost 30 years of doing research on HIV prevention in women, it is deeply disconcerting that we have been able to make such good progress on HIV treatment, but that has not been mirrored with progress on HIV prevention. Indeed, when we look at the targets that we had set, and I was part of doing this some 6-7 years ago, we had anticipated that we should be reaching around 500,000 new cases in the course of last year. But instead, we had about 1.7 million new cases. We are really not doing well. We are lagging far behind in prevention. So we've got to try and do better. So how might we do better? Let me share one new technology that we are involved in. Back in 2012, we looked at some of our cohorts to see if any of the women in our studies had broadly neutralizing antibodies. And we were very fortunate to identify someone who had a very broadly neutralizing antibody. Her research number was CAP256, and so the antibody is called the CAP256 antibody. We were able to isolate this antibody, and we were able to grow it in tissue culture, and then we tested it in animals. And in testing it in animals, it gave us a clue about its potential as a PrEP agent. So in this study, undertaken by our colleague Dan Baruch at Harvard, in the 9 monkeys that received the sham, all of them became infected when challenged. In the 12 monkeys that received differing concentrations of CAP256, none became infected. So that gave us some idea that this antibody, CAP256, is effective in monkeys in preventing HIV infection. So now we are developing it as a 6-monthly injectable. Can we provide women an injection once every 6 months that will protect them in those 10 to 12 years of their young period when they are at highest risk? Because women go through different stages in life and their risk is short-lived for that sort of period. And if we can provide them with protection over that period through this antibody, we can make an impact on the number of new infections that are occurring. So to date, we have manufactured the product under GMP conditions. We have completed the phase 1 studies, and we are now proceeding to the phase 2 studies. And this for us provides us one indication of how we can use technologies in order to empower women to protect themselves against HIV. So let me conclude with that reducing HIV rates in young women remains in Africa a key to controlling the global epidemic. 70% of infections occur in Africa. We've got to make a dent on new infections in Africa. Age-disparate sexual partnering is a key factor driving all of this. And genital inflammation is a biological risk factor that we've got to understand. Tanofeva-based PrEP is one mechanism, and it's being rolled out right now through oral PrEP that's widely recommended for women. But there are many challenges, and among those challenges, we have to deal with the biological basis on which tenofovir is being broken down in the genital tract. So we need longer acting new prevention technologies, ones that are not only dependent on antiretrovirals, and the ones that are not only impacted in this way by Gairdner vaginalis. And so we are developing a 2 antibody, both of them broadly neutralizing activity as a 6-monthly passive immunization, because for us it is a high priority that new HIV prevention tools empower women to protect themselves. We trust you found this presentation a journey over many years to give you some insight into the challenges we have faced in conducting research on HIV prevention in women, largely in South Africa. It was made possible by an amazing group of collaborative researchers, people we have worked with over many years, in laboratories, in the field, in the communities. And throughout, it has been a privilege for us to have had this opportunity to work with all of them. We'd like to thank them, our collaborators, our sponsors, and mainly we'd like to thank the many participants who gave freely of their time to participate in our research.
I'd like to also thank the large teams of scientists that have contributed to this body of knowledge. And let me end by again thanking the Gairdner Foundation and the Consortium of Universities for Global Health to have this opportunity to share this very stimulating, challenging journey with all of you today. Thank you so much.
Thank you, Quarraisha and Salim, for a fantastic talk and a fantastic journey that you shared with us through your goal of really trying to prevent HIV in Africa, but also prevention as a worldwide goal for this terrible disease. Those of you who would like to ask questions, I have a couple of questions coming up in the Q&A. Any other questions that people would like to put to Salim and Quarraisha? We do have some time for a Q&A. So if you'd like to put them in the Q&A box, I will pick them up there and pass them on. The first question comes from James Hudspeth, who says that he's very excited, as I think all of us are, about the possible passive antibody use for PrEP. And he's asking about cost and how you see this being, potentially implemented on, obviously on a large scale at a low cost. And that's a key question, obviously.
Yes, thank you very much, and thank you very much, James, for that question. We've been trying to understand what's the best approach to using broadly neutralizing antibodies, and our focus has been to use 2 broadly neutralizing antibodies from different classes. In our case, VRC07, which is an anti-CD4 binding site, and CAP256, which is a V1-V2 loop antibody. Together, they cover about 99% of clade C viruses and about 97% of other clades. So there's good coverage with that combination. The PK data suggests that we can use it roughly at about 6 monthly intervals. If we are using it at 6 monthly intervals, we can do so subcutaneously, and that would lower the cost very substantially. And the concentrations that we've been able to achieve mean that we would have to administer something in the region of between 15 and 20 mils every 6 months. The cost of that, at just a back of the envelope calculation, it works out it's not actually that much more than taking a daily pill of Truvada. So it's comparable in terms of cost, because you're only talking about 2 injections each year as opposed to taking 365 tablets. Critical for us, of course, is that as we improve the cell lines and we get a higher yield, we will be able to lower that cost. So I think at this stage, working with a cost of in the region of around $80 to $100 per year is about target that we're aiming for.
Thanks. I think that clearly gives us a goal to be shooting for, and one that makes some sense. Next question comes from Peter Kilmarks, who says congratulations and a great talk. Is there evidence for or interest in the use of HPV vaccination to prevent HIV? Is there a crosstalk there?
Thanks very much, Peter, for your lovely message of congratulations. So glad to have you joining us for this session, and great question. So yeah, we, through our analysis and other analyses done in Africa, we have found that HPV infection is quite common and acquired very early in young women. And some countries have started rollout of HPV vaccination in 9-10-year-olds, mainly as a mechanism to prevent cervical cancer. And we think it's a very great opportunity to extend that time period of vaccination from 9 and 10-year-olds to up to about age 20. And we could potentially have a dual benefit, both for HIV prevention and also for preventing cervical cancer. Unfortunately, we've not been able to get traction for this idea of this extension. And I hope that people listening to this talk and potentially some donors on may have some interest in supporting this, because I think this is something that's there, it's available, it is being rolled out, and a potential of getting more for the buck in terms of HIV and cervical cancer would be this timeline extension. Thank you.
Well, I think that goes in with the question that Keith Martin just put up on the Q&A. I think you've basically answered his question, where he says, more than 300,000 women die each year from cervical cancer. Can the HIV/AIDS community collaborate to vaccinate more girls and boys with HPV? And I think your answer is yes, but it's...
Of course, and we should. And I think with COVID, what we're seeing is we're dealing with multiple pandemics, and the more we can synergize our efforts between and among these pandemics, and HPV and HIV is an obvious one. There's HIV and TB as another one, and yes, we should stay focused. In a global health conference is a good reminder about the many pandemics and challenges that continue to face, that we face, and that we have opportunities to alter trajectories on when we work in a synergistic manner.
May I just add one small issue? I think that when you look at HPV in our research, we focus on the 46 serotypes that we have in our assay. So when you look at HPV occurring and clearing and persisting, the vaccine itself just focuses on the cancer-causing types. It doesn't look at the full spectrum. So right now, the vaccine looks at about 8 of the 46 serotypes. But there's cross-reactions between several of them. So a vaccine would make an impact, but a vaccine is not designed to stop all of the different HPV. So it's a bit more, it's not equating. You vaccinate somebody with HPV, they don't get HPV anymore. It doesn't work like that. They just don't get the cancer-causing serotype. So just raising that issue.
Okay. So the next question is from Teresa Townley, and it's more, I think, on the behavioral aspect, where she asks, what can be done about trying to reduce this age difference in HIV acquisition in young women? I think that's obviously a broader question that's societal as well. Quarraisha, do you want to try and address that?
Yeah, and biology. And when we think about this age-sex difference of about 8 years, I think with the technologies, what we're trying to do is deal with the fundamentally what is a social challenge. And underlying this is gender power disparities. So, to address this question needs us to bridge the gap that we have in terms of what underlies women's ability to protect themselves and how they can protect themselves. And the technologies are just one mechanism to do it. So we can try and change the behaviors by focusing on men at a younger age. So many of the interventions that we have start looking at the risk in men after about age 25. But to change norms, you've got to start working with men at a much younger age, 15 to 19. There's opportunities to do medical male circumcision there. But more importantly, it's about what is responsible behavior, what is masculine, what is non-toxic masculinities, and how to reach those issues so that women can survive to age of majority when they have cognition, to be able to make those choices and decisions at the right time that ensures they remain uninfected and can realize their full potential.
Yeah, thank you. So Judy Wasserheit asks or comments, the one ongoing challenge has been prevention and cure of BV. I'm not quite sure, BV. In the light of the data you presented, this is potentially important for HIV prevention. Can you comment on approaches to this challenge?
Sure. Thanks very much, Judy. It's wonderful to have you join us in this session. It's a long time since our paths have crossed in relation to COVID. Just to say that I think our understanding of the microbiome has been growing in leaps and bounds over the last few years as the technology has improved, as our ability to sift through different aspects of the microbiome, we have a better understanding. And I think, like you, we've really grappled with what is BV. It's not a clear-cut causation. We don't really understand what's underlying it, except that it's a dysbiosis, and it's where the expected flora have been altered in some way, and that's what's negatively impacting. And that's when we start seeing Prevotella and Gairdner taking over. So the big problem has been, how do you treat BV? And we've grappled with this in many different ways. Of course, we treat with metronidazole, which clears it up, but then 3 months later it's back. we don't really have a way in which we can treat it and ensure that it remains a cured condition. And so I've been very excited to see some of the new results that's coming from the Lactobacilli crispatus studies that show that if you treat with metronidazole followed by putting in a lactobacilli, that we can alter the microbiome. But even then, it only applies in about half the women. So we still have many challenges. And I see these as sort of baby steps on the way to really grappling with BV and moving us to a position where we can alter the microbiome, that it will remain consistently in a much more desirable characterization than the diversity we see in BV.
Okay, so Keith has a comment which you can read. Griffin Barris, again, congratulations. And back to the sort of social elements, and an interesting question actually, whether there is a stigma associated with HPV vaccine in sub-Saharan Africa, particularly parental concerns that vaccination will encourage sexual promiscuity, because this is, and it has been an issue in the USA. And do you see this potentially as a hurdle as you think about antibody treatment as a prevention for HIV, the social acceptability of this? Quarraisha?
So, yeah. So I think, the issue of anti-vaxxers is not as big an issue in South Africa as we've been noting in other parts of the world. And part of that, I think, stems from the fact that many parents have seen the benefit of childhood vaccinations in terms of child survival in Africa. So I can speak mainly in South Africa. We have high rates of uptake of the childhood vaccinations to a point where if there are shortages, mothers will go to extreme lengths to find clinics where they can vaccinate their children. And we've only fairly recently introduced the HPV vaccine, and it's because we don't have enough, it's an issue of affordability and it's being introduced. It's a very narrow range that we are offering it in 9 and 10-year-olds, mainly through the school system and largely through the public sector. We've not actually encountered much resistance in terms of HPV vaccination. I think there's also been campaigns, annual campaigns, where there's a lot of attention drawn to cervical cancer, which is fairly common. And so people see this, the vaccination, as part of the solution in terms of preventing cervical cancer. But there are small groups that do express concern, but fortunately they remain fairly small and not having such an impact in terms of lowering rates of vaccination coverage in our setting. Thanks.
Thanks. So I think we've covered a lot of ground on HIV. We can't end without asking you about COVID, because obviously both of you, particularly Salim, you've been very active as a voice of science in South Africa around the COVID pandemic. How do you see South Africa's response? It seems as though you have been able to develop a good response, but what is the access to vaccines? Is COVAX good enough for you? Can you get vaccines into South Africa?
Yeah, thanks, Janet. Firstly, let me just thank everybody who has joined us here today, and want to just particularly thank Keith Martin, Stephanie, Evelyn, Judy, Marlene, and Peter Singer, and many others for their wonderful comments. Really, really deeply appreciated. When we had our first case of COVID back on the 5th of March last year, just over a year ago, I don't think we really understood what we were dealing with at the time, but the president took very quick action. Within 10 days, we instituted a state of disaster, which helped close the borders. It reduced prohibited gatherings. So there were a whole lot of steps that were put in place. And shortly thereafter, we went into a lockdown. And what that effectively did is that it took an epidemic that was growing at the same rate as the UK epidemic, doubling every 2 days, and we flattened that curve. So it became one that started doubling every 15 days. So community transmission really slowed down due to these interventions. But we knew that as we eased restrictions, it was going to come back, and so it did. But when it came back about 8 to 9 weeks later, and we had that opportunity to, get enough oxygen, make sure the field hospitals were built, put our testing into place, and so on. So we did all of those things, which made our first wave somewhat less, severe than we were expecting. But the second wave, we were not expecting a new variant, super spreading events when young students from universities and students were going to have parties at the end of their final examinations. And before we knew it, we were in the midst of a really severe wave. And that came because of this new variant. Now we are in the post-second wave. Cases are low. We're in that honeymoon period. And the cases have come down, partly because of government action, partly because of people's changing of their behavior, and partly because of accumulating immunity. So all those things have come together to enable us to get back into a situation where, restrictions have eased.
And vaccine accessibility.
Oh, I was going to talk about vaccines. Yes, vaccines have proven to be a huge challenge. I mean, we committed ourselves to COVAX. South Africa has been a very big supporter of vaccine equity. But because of the huge political pressure in South Africa, our government was put into a situation by, sadly, many scientists and others of going around COVAX and jumping the queue, paying higher prices, buying vaccines, doing anything to get vaccines. And I kept saying, well, if I'm a pharmaceutical company, I would really like to have 50 different countries coming knocking on my door to buy my few vaccines, as opposed to them going to COVAX and saying to COVAX, can you buy us the vaccines as a single large purchase where you can negotiate better prices? So we have, in a way, shot ourselves in the foot in the way we've acted. But I understand people want a vaccine. And I've been saddened in some instances when I've seen the kind of Trump-like, me first, I want my vaccines, I don't care about others. And I'm saddened to see that. But I'm hopeful that as vaccines now become available and COVAX, which was very slow off the blocks in supplying vaccines, is now addressing that. And so now we're busy vaccinating in South Africa, and it's in fact now in something like about 17 different African countries that have started vaccinations.
So that is good news, but of course, vaccine nationalism is widespread. So you are certainly not alone, and this is going to be an interesting year as we move into broader accessibility of vaccines, because as you and others have said, it's not enough to vaccinate the North. We have to vaccinate the world if we're going to. I think at this point, it's time to say thank you very much to both of you for a wonderful talk and a good discussion. And congratulations again. And we look forward to seeing you in real person at some point during the year ahead. So thanks very much. And thank you to the audience and to Keith and everybody at CUGH.
Thank you so much, Janet. And thanks to all the participants for joining. Thank you.
Thank you.